preparing to go live. That's it.
And where is Fatima? She's there.
She's there.
Okay. Another 1 or 2 minutes. Start. Please start. Sarin, please take some photographs also.
Okay, ma'am.
So how is the weather in Kochi? Is it raining now also?
Yeah, morning it was raining a little bit, but at the moment it is very cloudy, but not raining.
Because Delhi also had lots of rain last 3 days.
Okay.
But since yesterday it's okay. Maybe just a spell, and the next week we are expecting again more rains.
Okay.
Good morning. Good morning, Dr. Susan.
Rainfall.
Good morning, Paranjit.
Hi.
Paranjit is in our executive committee. She was earlier principal of Bhavan's College, right?
Yes.
Yes. Yeah.
Okay.
And head of microbiology.
We share the same name. I'm glad to see I share my name with someone else.
Yes. Okay. Shall we start, ma'am?
Yes, you can start.
Fatima?
Yes, ma'am.
You can take over.
Okay. Ma'am, so is it audible?
Yes.
Okay, ma'am. Good morning to all the respected dignitaries, teachers, and my dear friends. It's my pleasure to welcome you all to an interesting webinar session. Hope you all are good and safe. Thank you for taking your valuable time out and being here with us from all the world around. Department of Bioscience, Union Christian College, Alua, Kerala, proudly present to you this webinar on the topic Climate Smart Agriculture in the Era of Plant Genomics by Dr. Paranjit Khurana, University of Delhi, as a part of our ongoing centenary webinar lecture series. We have with us former BARC scientist and adjunct faculty, Dr. Susan Epen ma'am, Dr. Rita Mukhopadhyay, President of Indian Women Scientists Association, Head of Department of Bioscience, Mr. Shyam Mohan sir, Faculty Coordinator of the program, Dr. Sareen Sarah ma'am, and other faculty members of UCC. And I heartily welcome our Principal, Dr. Thara K. Simon ma'am, and Reverend Thomas Jones sir in their absence. Before we begin, let us invoke the Almighty's presence and blessings in this gathering with a silent prayer.
মিনিট 10-এর জন্য একটু ব্যস্ততা আছে, তারপরে পরমজিৎ খোরানা ও আজকে কথা বলবে ক্লাইমেট চেঞ্জ।
Before we get started, here's a gentle reminder. Please follow the instructions during the webinar for the smooth functioning of the program. Be sure to mute your audio so that there is no disturbance during the session. I request you all to mute your audio. If you have any queries, please type those into the Zoom or YouTube live chat box. You may send in your questions at any time during the presentation. The feedback form of the webinar will be available at the end of the program. So moving ahead, I now invite Mr. Shyam Mohan Sir, HOD of Bioscience, to welcome our respected dignitaries with a welcome address.
Thank you, Fatima. Today we have a seminar that is hosted by the Department of Biosciences in collaboration with the IWSA and the Board of Nuclear Energy, DAE. So today's topic for the day in our webinar is climate smart agriculture in the era of plant genomics, and our resource person is Dr. Paramjit Khurana. I take this opportunity to welcome the Chief Resource Person of the day, Dr. Paramjit Khurana, who is a scientist in the plant molecular biology in University campus of New Delhi. Firstly, I would like to welcome the speaker for the day, Dr. Paramjit Khurana, especially a relevant topic, just like we were discussing about the scanty rainfall and sometimes heavy rainfall. So the methods of research that can be input into the field of plant agriculture using technologies in genomics, etc. Welcome, Dr. Paramjit Khurana, to this gathering as the Chief Resource Person. Now, I would like to formally welcome the Principal, Dr. Tara K. Simon, and the Manager, Reverend Thomas John Achin, who in their absence will help us be motivated and for the smooth conduct of this seminar. I welcome Dr. Tara and Reverend Thomas John Achin. I would like to welcome Dr. Susan Epen, the prime motivator behind the conduct of these seminars. And for the last 1.5 years during the time of pandemic, we have hosted a lot of programs under her guidance. Thank you for your constant motivation and support, ma'am. Dr. Susan, ma'am, I welcome you. This seminar is coordinated with the help of IWSA and the Board of Nuclear Sciences. So all the dignitaries, Dr. Rita Mukhopadhyaya is the president of the IWSA. She has been here in our college. I welcome her as well as all the dignitaries of and the those who constitute the memberships of these organizations to our webinar. Welcome all of you, ma'am. I would like to welcome the faculty coordinator of this program who conducts volunteers, the students, and motivates every week. We have at least one seminar, and Dr. Saren Sarah John is the guiding light behind the program, the different programs that are conducted with scientific perspective and academic interests. Saren ma'am, you've been doing a constant good work, and I welcome you to this program. It's also my pleasure to welcome the faculty and non-teaching faculty of our college, and who, especially Department of Biosciences. Each of us are waiting, and we have had different topics altogether. And today is also a diverse topic that we are engaging in. So your constant help in the conduct of these programs is invaluable. So I welcome all the faculty and non-teaching faculty of Biosciences Department and Union Christian College. I also take this opportunity to welcome all the participants of our seminar who are tuning in from all over India. And your presence is very much needed for the success of all of our seminars. And you've been constantly deliberating it through participation. Welcome all the dignitaries, and a special word of mention to welcome all the student volunteers of our department who are engaged in the smooth conduct of this program. Thank you. Welcome one and all.
Thank you so much, sir. I take this moment to welcome you on this behalf of this gathering. Next, I call Dr. Rita Mukhopadhyay, President of Indian Women Scientists Association, to speak a few words about the activities of IWSA. Welcome, ma'am.
And good morning to all the listeners, audience, students from the college and from the faraway universities who have joined today's meeting. We have a very important speaker, and we are all looking forward to hearing about the topic, which is the crux of the time. But it is my pleasant duties to also take away a few minutes from this session to talk about the organization which we all stand by, the Indian Women Scientists Association. and you see the logo of IWSA, hands holding the atom, and we have our speaker, Paramjeet. So we are all connected through the matters and molecules. Here on the left are the founder members whose vision we are fulfilling and taking forward this institution to the next 50. Yes, we turn Golden Jubilee year from 2022 June, and we want to have a celebration like how the Biosciences Department of Alua College is happening on their centenary. So we wish to follow in the footsteps and celebrate in various forms of scientific deliberations and collaborations. On the right, you see the Board of Trustee members. They are our guiding and immediately abiding sources of all the activities which we do. And the slogan is, yes, indeed, the slogan for our Golden Jubilee. Looking back to look forward, soaring high with the winds of change. And that's what IWSA has been following, practicing, taking it forward to the society with their scientific mindset, scientific illumination, and more to come on the way as we interact with more people through the network. So did the COVID time really bog us down? No, it cannot, because that is what the next slide will tell you. This is of course only a glimpse at all the community welfare activities which we have. And currently the pandemic has affected our nursery school, the daycare center, the healthcare center, which has started to come back again into action. But the other places, like the Working Women's Hostel, for a single day, it did not close its door. And now we are again seeing the increasing footfall of those women who come to Bombay to pursue their careers. Computer Center, I will talk about it at the end, and it is functional. We have the 25th year celebration on a diploma course, which is open to, again, Pan-India. So I put it on this slide for anybody aged between 18 to 50 can enroll for this program and get the benefit of empowering themselves from nursery or daycare teachers. We have an advanced learning center, which again asks students to become computer savvy. Of course, in today's day time, everybody has a mobile phone in their hands. The government has also provided the school students with these devices, but we are open to even teach those who are still not having those opportunities. We have scholarships and awards, and students are encouraged to look up the website so that at the time of need, we could be of help, of a small assistance may it be, but still it's a relationship. The next slide. We want to make a slogan of restore, recreate, reimagine, very relevant to what today we are going to hear from Dr. Paramjeet. And at the IFSA, I only wanted to take this slide as a cue to say that yes, we reimagined. We became compliant to the safety measures and have kept the hostel open. We have recreated our brain wiring, and instead of meeting in person, we now meet on the virtual platform. And can you imagine if I say that from 9 to no end to the time in the evening, most of us are engaged on our laptops working from our home for the institute, for the sciences, for taking to the society the message of restoration, may it be scientific deliberations like these, or may it be workshops on the latest of technologies. Next slide. We'll browse through these only to tell you what, the next slide, please. It only shows you that our hostel is compliant and is, it's a beautiful place. We have a garden which we have developed into a learning garden living museum. It's going to be inaugurated for our Golden Jubilee coming year, with a wall mural and our slogan and many more things which can be picked up on the way of this next 10 months. And that is our hostel. This is our garden. This is our compliance to COVID safety. Next one, please. As I said, the healthcare center, Yeah, I have already spelled on this, and you can see that during the COVID, man was affected and nature was spared. And that is why they came in bounty. And this is only a glimpse of what happens during the seasons of the butterflies and the seasonal flowers of variety of colors and their coordinations across our museum garden. Only a glimpse. Let's hope that for our conferences, you all will be present in the coming year. Next slide. Yeah, this is our activity center for the service to community through our health sciences. Next one. I will quickly go through it. Yeah, green initiatives, and the one on the left you see has developed during the pandemic year. And now from this month, it is giving us its energy. Next one. Yes, what did we do otherwise as well? We bridged the gap between society and the funders. If there is no funding, we cannot go ahead. So the new programs of collaboration opened up, and Vigyan Prasar has become our one collaborator who has supported 3 projects. One of it being Each One Teach One, another one being the Learning Garden Museum, and the third one being the Vigyan Prasar network of students all across Maharashtra. Next one. Yeah, this is only a picture on the wall to tell you how many new programs we had begun. And we have, now I'll talk about the next program which we did. Our voice is to go through lectures. And the next slide will show you that we initiated a few more lecture series during this pandemic confinement to our home lives. And member enrichment program, where members used to talk and discuss about their sciences and their capacity building workshops. We brought in Science and Our Life series of lectures where eminent people of the society speak to the society and convey the message of how science and imbibing science in their daily life is important for us humans as one more species on this green planet. Member empowerment programs, and wellness, health, and happiness program. Yes, again, I'm always going back to the pandemic, not to remind you of the tragedy which it has created, but only to tell you and give you we want to share this shoulder to shoulder, that we have to learn from what we get and evolve to adapt. That's what we do, adapt and go forward. So this is only why I'm making a reference all the time. So wellness, health, happiness is very relevant, and we have started this program as well. Next, I will talk about quickly on the latest 2021, and that is internship programs. And students and audience on this session may take a note of this, that we have already done 2 programs of 53 to 54 students in average over a program of 1 week to 1 month. And we are doing all the sciences to be covered in these internship programs. And you all can apply through the colleges, or you all can apply independently. On the left, you see a program out of the 10, which was nothing but restoration of the mangroves initiation, which we have done. And that became successful because our interns could nurture the saplings in their own homes, grow them up to a sturdy self, so that we could take it back to the coast. And this is only a beginning, and we want to join hands with the network. Next one, please. A glimpse of all the other 10 programs, and the programs vary from using Python language to establish science quiz or any other aspects of science they want to cover, to microgreens, hydroponics, mushrooms on alternate substrates, and may be many. You are requested to please go to iwsa.net to update yourselves on this. And we are soon going to begin the chemistry and physics internships. And again, another college is lining up from November to do a biotechnology internship. The last slide is almost there. I want to talk about, this is our voice. We have a newsletter. Again, it is now in the e format, available at our website under the publications. And this comes up every 3 months. and it gives us a record keeping of all the activities which we conduct. So please do browse whenever you have time and contribute towards it. And that's how we take science to everyone, for everyone. And the final slide is that our computer center, which started 30 years back, can you imagine? Those founders had already thought that computations is the way of life, and that's what we are living. And this center is turning 30, and our first program is coming up. The next slide and the last one. The next program is coming up for the celebration of this event. We are organizing a half-day international lecture seminar with eminent speakers, Bhatnagar awardees, Padma Shri awardees, to speak about how the digital era evolved and where we are today from where we had begun. So with this, this is the first flyer. I should encourage all the student participants to look it through, and please do come and join, participate. That is the email where you can show your interest, and let us all work together for a better tomorrow is the message for today, when we look forward to hear from Dr. Khurana how the plants around us should change for the time to support the most selfish species on earth, the humans. Thank you. Bye.
Thank you so much, ma'am, for giving us the glimpse of the association and the insights of your program. Next, I welcome Susan Epen, ma'am, to introduce our guest speaker and presenter of today's webinar session. Please, ma'am.
Ladies and gentlemen, it gives me great pleasure to introduce Dr. Paranjit Khurana, J.C. Bose National Fellow, Department of Plant Molecular Biology, University of Delhi, South Campus, New Delhi. Dr. Khurana did a BSc, MSc, and PhD from Delhi University. She is a fellow of all the science academies like Indian National Science Academy, New Delhi, Indian Academy of Sciences, Bangalore, National Academy of Sciences, Science India, Allahabad, National Academy of Agriculture Sciences, and Third World Academy of Sciences. She was the Foreign Secretary of NACI. She is a member of several organizations, member of Third World Organization for Women in Science, Italy, International Agriculture Commission, France, Society of Plant Biochemistry and Biotechnology, Indian Society of Developmental Biologists, Indian Cell Biology Society, Indian Science Congress Association, Association of Plant Tissue Culture, India, National Academy of Sericulture Sciences of India, Bangalore North Zonal Chapter of NACI, etc. She is also a member of several governing bodies like Institute of Home Economics, University Hostel for Women, College of Nursing, and Netaji Subhash Institute of Technology, all of University of Delhi. She is a member of Star College Advisory Committee of Maitreyi College, Ramjas College, and Miranda House of University of Delhi. She is a member of Special Committee of School of Environmental Science and School of Computational and Integrative Sciences of JNU, New Delhi. She's a member of Academic Council of several places. She's also a member of Task Force of Human Resource Development, Department of DBT, then Ministry of Science and Technology, Government of India, then Silk Biotechnology, Department of Biotechnology, Ministry of Science and Technology, Government of India, Biocare, Department of Biotechnology, Department and other for value-added biomass and products from natural resources, etc. She's a member of National Bioscience. She is a member of National Bioscience Award for Career Development in 2011. Her research interest varies from plant genomics to development of transgenics to development of climate-proof crops, etc. Her major work includes pioneering contributions in the area of wheat and cereal biotechnology and comparative genomics. She has done extensive work on genetic transformation of Indian wheat, especially for resistance against cyst nematode and abiotic stress tolerance. She's a specialist in mulberry transgenics, capable of withstanding salinity and drought. And she's also interested in stress tolerance in changing climatic conditions. She has she has also worked on molecular basis of somatic embryogenesis and heat stress tolerance in wheat. She has sequenced chromosome 11 of rice, chromosome 5 of rice, and as a part of the international initiative. She has sequenced the chloroplast genome of mulberry. Then at present, she is involved in sequencing of chromosome 2A of wheat. She has more than 150 publications in international and national journals. She has presentations in conferences and workshops, more than 200. She is in the editorial board of Journal of Cereal Biotechnology, Physiology and Molecular Biology of Plants, Proceedings of National Academy of Sciences, Section B, Biological Sciences. Then she has also received several awards, such as Professor Archana Sharma Award. Then she has received Sri Ranjan Memorial Lecture Award and Bharat Ratna Rajiv Gandhi Mahila Shakti National Award, Birbal Sani Award Medal, and several other lecture awards. It's my pleasure to welcome Dr. Khurana, who is just going to give her a talk on how to develop stress-tolerant plants in the era of genomics. Dr. Khurana, please.
Thank you so much, Dr. Susan, for a very elaborate introduction, I must say. And many thanks to the Department of Bioscience for inviting me, for giving me this opportunity. Thanks very much to Indian Women's Scientists Association, which I've been a member, and I've known many of your executive members like Sunita Mahajan and others. I've met them at various forums. It was always a pleasure to visit the premises at Vashi, Mumbai. I've been there a couple of times, and I've really appreciated very much the infrastructure which you have created there. So thank you very much again for inviting me and giving me this opportunity, I would say. I would like to start my screen share if I'm given that permission.
You can.
Yeah, it's not coming, but just a minute. Yeah. Okay, so the topic which I've chosen for today's presentation is climate smart agriculture. And in a while, I will tell you why I talk of climate smart agriculture. Because we are living in a very technologically advanced timing where we need to integrate the technology so that the classical agriculture can become climate smart, okay? So that it becomes adaptive to the changing climatic condition. And couple it up with the era of plant genomics where we are getting lots of newer information in plant sciences. So I wish to integrate the two, and I will try to take you through some of the work which we have been doing at the Department of Plant Molecular Biology at University of Delhi, so that the students and the others can relate to how we are addressing these problems with our local resources. Okay, so what do we mean by climate smart agriculture? I think this is the changing face of agriculture world over. You see agriculture not being done in the classical ways, but agriculture being done in a way where you're efficiently utilizing your land resources, you're utilizing the water resources and the bioresources in such a way that irrespective of where you are, you can utilize this so that you can manage depending upon the ecosystem in where you're present or what the ecosystems are, you can head towards a sustainable livelihood. The key, I think, depends upon these three aspects as to how well you're able to manage your land, water, and your bioresources. That would be the key towards. Now, coming to this, on the other side, we said a while ago that we are in an era of genomics. Now, to put it very simplistically, we knew about DNA making RNA to protein and metabolites, and all of a sudden we have now shifted to genomics, transcriptomics, proteomics, and metabolomics. What do we mean by that? Well, when we're talking of DNA, we're talking of one fragment at a time. But when we're talking of genomics, we're talking of the entire genetic complement of that organism. Similarly, when we're talking of RNA, we're talking of one transcript. But when you're talking of transcriptomics, then we're talking of the entire transcriptomic profile of either that organism at that stage, in that tissue, and so forth. Similarly, from one protein, we are not talking of proteomics. That means the entire protein complement of the organism which is there. And same goes for the metabolite also. Now, why we are doing this? While we have moved from here to here, what new information do we get out of it? So when we're talking of genomics, we get an inkling of what can happen. When we're talking of transcriptomics, because we're talking of RNA, we know what appears to be happening, what is the real factual situation in that tissue at that time. And in proteomics, what has happened and what made it happen. Okay, so you can address different aspects of this living cell and get information in different levels. Back home at Delhi University, we were very fortunate to be part of the international rice genome sequencing effort. And like Susan was saying, we were part of the international sequencing, and we did a part of chromosome 11 of rice. Subsequently, we have also been part of the international tomato genome sequence, and we've contributed towards that. We have also finished the wheat 2A physical mapping and the sequencing also a couple of years back. While these three were international collaborative programs, standalone, we focused on Mulberry. I'll come to Mulberry in a while. This was a home alone project. I mean, no one contributed to it. It was totally Indian effort, and we did that. But having done these genome projects, we all wanted to know how we can utilize that, and we moved over from the genomics to the transcriptomics also of wheat and mulberry for different stresses. And recently we have completed the mulberry draft genome also. We have sent it for publication. So we should be able to, this is again a standalone Indian effort. So we've been part of various plant genome programs at Delhi University. And that is what I thought I will take you from. Like, this was the Indian Initiative for Rice Genome Sequencing, where we were assigned chromosome 11. Chromosome 11 is rich in disease resistance genes, and it is only this long arm, which we were given to sequence together with colleagues in IRA. At UDSC, that is University of Delhi South Campus, we sequenced about 8.9 MB at that time, and this was with the older DNA sequencing methods, that is the Sanger's method of sequencing, and we were able to accomplish about 1.16 centimorgan region of the chromosome of rice. Now, this information of the map-based sequencing we published in 2005, and here you can see here how the 12 chromosomes of rice are distributed and how different countries contributed towards them. Chromosome 11 is the one to which India contributed, and interestingly, every chromosome makes up one molecule of DNA, one large pseudomolecule. And we were new entrants in the international arena at this time, yet all the 12 rice pseudomolecules were assembled in-house at the University of Delhi in a small lab, which we had set up for this purpose. Why I'm mentioning you must get the information from all countries with a constant parameter. No variables anywhere, only then you will be able to assign it. That means international standards had to be monitored, and that is where, when this work was accomplished, all the PIs from China, from in the USA, from China, from Korea, Japan, Taiwan, and others, they assembled together here. We have our Indian representative, but I do want to mention this person here, Masaki Furuya, who took a lead in this project. And Japan, you will see, sequenced about 5 chromosomes, contributed towards 5, not complete, but towards 5 major chromosomes. So this genome, usually, although we had published the sequence in Nature, and there is always a rivalry between nature and science never covers that issue. But for a change, science covered this because this was considered as a landmark development in plant sciences. And till today, this genome sequence is considered as the gold standard quality. This gold standard quality is given when the error rate is extremely low. There is variation of only one base pair allowed in about 10,000 base pairs. And that is why next to Arabidopsis, it is only rice which has the gold standard, and no other plant genome till today has achieved the gold standard rate. So having contributed to the rice genome, we wanted to do something on our own. And since I was already working on mulberry, I will tell you later. So we took up the challenge of sequencing a complete chloroplast genome of mulberry so that we could assemble this entire genome which is available. And fortunately, we were able to do that. And we published that in 2006. We used this information, but looking at our credibility in the international arena, we were invited to participate in the tomato genome sequencing also. Now, why tomato? Because tomato is a fleshy fruit. And if you have the genomic information of tomato, you would get a peep into how the fruits have evolved. And that is where the Tomato Genome Consortia invited India to participate, and we did participate and contributed to it. And the sequence of the tomato genome was published in 2012 here. Now, in case of tomato, you will see that although there are more than 7,000 varieties which are cultivated worldwide, we found out at the genomic level that there is very little genetic variation between them. And the reason is the difference is more apparent in the transcriptome and the epigenetics also as to how the different genomes have evolved here. So it gives you good information. And slowly, this tomato genome consortia transformed into Solanaceae genome program. That means the entire Solanaceae family was being targeted. Why? Because you see that in the members of the Solanaceae, you have members which will grow in the dry desert to the cold desert. You have vegetables like the capsicum, the brinjals. You have medicinally important plants like this is Withenia. No, not Withenia. This is nightshade. Yeah, Withenia. You have horticulturally important plants like petunia, tomato, of course, and tuber plants like potato, sweet potato, capsicum, and so on. So at that point of time, it was envisaged that if you had the genome of one organism, that is tomato, you could extrapolate this information to the closer relatives of tomato as well. And that is why the Solanaceae genome project was initiated, so that we could systematically approach diversity as well as adaptation of plants within this particular species. We always were keen to work on the wheat genome. However, in the earlier stages, we were not very successful because wheat is a very complex genome. It has a hexaploid genome, and it was only later that we got enough funding for this. And in 2018, we published the International Wheat Genome Sequence also, and India contributed to the 2A sequencing effort in this direction also. Now, all these genomes sequencing leave you with a question which is very important to answer, and that is, what is the future of farming? How can you translate this high-end information into such a way that you will be able to help the billions of mouths to feed? And why this is important is, it is important because this is a very outdated diagram to show you that there is population explosion in the Indo-Western, Taiwan, China, in this peninsular region, covering these major countries. And every dot is about 1 lakh people here. And that is the reason that we have a bigger responsibility of feeding this growing population. So what do we do? The basis of food security is, of course, agriculture, and that is why agriculture needs to be integrated with ever-evolving technologies. And we've been able to do that successfully in India. Like, we started with the Green Revolution when the dwarfing genes were introduced in. We then went on to the gene revolutions, where genes were identified for crossing over, for genome-wide allele mining and other things here. And we are heading towards evergreen revolution if we are able to judiciously utilize the information which is available with us. Of course, the food security poses many challenges. The chief being that we are actually outgrowing our natural resources. What do we mean by that? I bring in a cover page of this book, which is very aptly titled as Outgrowing the Earth. And it says that the major food security is that the water tables are falling and the temperatures are rising. Very clearly put in by Lester Brown that the water table is going down, water is getting scarce, and the temperatures are rising. In our research at Delhi University, we've tried to address these two, and I'll try to give you a glimpse of how we'll be able to address these two projects with plants and crops of local interest, of interest to Indian population. Of course, the other book written by this author is that the planet is full and the plates are empty. It means that there is enough food for everyone, yet there is a politics of food distribution which is creating food scarcity. Of course, I will not touch upon geopolitics of why the plates continue to remain empty when the planet is full of lots of food for others. That's a different perspective, economic distribution and other things. I will not get into it. So today I will focus on these two major issues, the falling water tables and the rising temperatures. Water is very important. Okay, I believe water is more important than gold, diamond, or silver because it has no substitute. You cannot substitute water with any alternative. You cannot do without water. While our Earth has about 97% of water which is present, is not suitable for human consumption. And very little, only 3% is fresh water, which is we can use for human consumption. And of the 3% also, 68% is blocked in ice caps and glaciers. That means it is not available to you. What is available is 30% of this 3% is present in the groundwater, okay? And only 1% is the fresh water. And this fresh water is important because the fresh water is the one which is going to feed your agriculture. And it is agriculture alone which will be able to put the food on our plates in future. And when we look at the the satellite image of groundwater resources and what is the rate of depletion of these groundwaters, you will see there are three rectangles where the water tables are going down at a remarkably fast pace. And if I enlarge one of these is majorly concentrated in the breadbasket region of our country, that is wheat and rice, the Haryana, Punjab region, Rajasthan region specifically. And if you look at the color scales, the water tables are going down more than 1,000 millimeters per year. That is about a meter per year. And this is about good 10 decades earlier data. And the situation has not improved. It has gone from bad to worse only. The depth has increased. The spread of this scale has increased here. So what should we do when this is the situation? If water is not going to be available in abundance, then you try to restrict your methods and other things and try to avoid using water wherever possible. So I bring in this slide to show you that the corporates are highly conscious of this. The Levi's brand of popular American original jeans, they used to use about 42 liters of water in the finishing process per average pair of jeans, okay? Now they have developed a process wherein they can reduce the water processing by about 96%. I would say that is corporate consciousness, that because this water is going to be wasted. It is a processing water, and if you can have alternatives for that, that is a good thing. So you can do away with water where you can have an alternative. But in case of agriculture, you cannot do that. Certain crop plants need more water, some need less water, okay? All crop plants need water to grow. This slide tells you that corn needs about 450 liters to produce 1 kg of corn. Wheat about 500. Rice, 1700 liters to produce 1 kg of rice. In fact, the total virtual water content is actually more than that. For rice, you need about 2,300 liters as compared to wheat, which needs nearly half of it. And that is the reason in our agricultural practices, rice used to be sown in places where water was abundant, and wheat where the water was not that easily available. But with our economic pressures, now rice is being grown in places where wheat should be grown, and that is what is adding to the depletion of water resources. While it is very difficult to tell the farmers not to do this, not to do that, because this is a high-priced, exportable product, commodity, and gives you good returns. Everyone wants to grow rice, but it has a big penalty on the groundwater water here. Water is something which is going to be very dear to us. It is going to be the commodity of the future. This simple molecule, as I mentioned here, chemically, H2O. The plants, I think, are very smart. They regulate the water supply, depending upon the water supply, which also in turn is affected by soil texture, soil porosity, the root structure, how it is developed, the soil temperature, the depth at which the roots are going in, depending upon the availability in the soil and the atmospheric demands. And that varies. What is the temperature? What is the radiation? What is the rolling? How are the leaves getting it? Whether the stomata are open or not? What is the humidity level? How fast is the wind blowing? They regulate it very well. The plants are master them. So we thought, let's take a clue from the plants itself. And the clue which we took from the plants was the presence of certain special proteins which will help it tolerate against drought and salinity. This protein is known, the one we have taken is from Odium vulgare, agglutinin 1. It's a layer group of protein. And this protein binds to the membranes and maintains cellular structures more efficiently under these abiotic stress conditions of drought and salinity. A while ago, I said I also work on mulberry. And mulberry is grown in the southern region, and I'm sure it is more popular site in Kochi and nearby areas which have abundant rainfall. Reason being, mulberry is a very fast growing plant. It's the fastest growing dicot. Next to bamboo, mulberry is the fastest growing plant. And it thrives in these conditions, and it's a plantation crop. So once you plant it, it keeps on growing, and the farmer keeps on chopping the shoots and feeds it for silk production. Okay, so sericulture is totally dependent on mulberry cultivation, and mulberry cultivation the mulberry plant uses lots of water. So we were actually contacted by the Central Sericulture Institute located in Mysore to develop certain mulberries which will grow under less water. And at that time we had this HVA gene with us, which we are using for wheat, and we thought, let's try it on mulberry also. And to make a very long story short, about good 20 years of work, 10 years of work has gone in. When we transformed the mulberry plants with the HVA gene from Hodium vulgare, you can see this is the control plant. When you do not water them for a week, 10 days, this plant starts wilting, the natural ones, because mulberry needs lots of water. But the transgenics, which overexpress the HVA, they're still better. In fact, not only 10 days, but even till 20 days. What is important here is, if you look at the relative water content in these leaves and compare it with these plants, if you focus on the blue bar here, you see that the transgenics have about 75 to 80% of relative water content still available with them, while the non-transgenics, the water content has gone down to about 40% or so. So these transgenics were characterized, you could not water them. And at Mysore Institute, they did not water the plants for about 2 months or so, and the plants still survived. While the non-transgenics, they did not survive at all. These transgenics were characterized thoroughly in our greenhouses at Delhi, and they were subsequently transferred to Mysore in 2009. And later, we have about different genes which we introduced in mulberry also for drought and salinity, in fact 4 of them. And they were also transferred to the University of Agricultural Sciences, Bangalore in 2013. Okay. These transgenics have been growing well at the Bangalore University also, and they are thriving there also and are being characterized for field release if the government of India permits it at some point of time. Right now, we are stuck with the biotech policies and no such plant is being released here. Okay. So coming over to the other plant which we work on, wheat, as I said, wheat is important because not only is a food which feeds the world, it also is responsible for its healing. We worked on different types of wheat, durum, dicoccum, estivum, and so forth. They have different ploidies and they have different genomes as well. But today I will focus only on the hexaploid genome. Wheat genome, as I said a while ago, is highly complex. Its size is huge. It is 16,000 MB. It's a hexaploid, 6N ploidy level. And if you look at rice, it's about 430 MB or so. And I often joke that as if we don't have enough variation in the world with two parents and all that heterozygosity which you see in and around you. In case of wheat, it has three parental genomes, A genome, B genome, and D genome. Okay, so there is so much of variation here. From the molecular point of view, nearly 80% of the DNA is repetitive DNA. So when you want to do any genomic work, it makes hell of your job, hell of a task here in this. And of course, it is susceptible to abiotic stress, and it together with rice, it's an important crop plant here. And when we started this work in the early 20s, I would say, on wheat, we had lots and lots of difficulty in this particular. We developed our first transgenics of wheat and durum. That is what the cereal cyst nematode one was, and subsequently in others in dicoccum and styrone. But it is the bread wheat which we were after, and to have it in bread wheat, we utilized a different approach. That is to create first haploids, transform the haploids, and diplodize them. Then you have doubled haploid. So it's not one genome which has got doubled, but the entire genetic complement gets doubled. Why do we do this? This is because here now the gene which we have introduced becomes stable. And to do segregation analysis in bread wheat is difficult. And if you have double haploid, then you don't need to do that because that does not occur. And we have characterized these double haploids. Now in here, we had introduced in the gene for HVA, the same one I'm taking today, showing you the data only of that. And if you see at higher stress, when you give simulate drought stress and you give it by giving mannitol or other things here, the non-transgenics do not germinate at 300 or 400 millimolar, while the transgenics regenerate very well as compared to this. This work we published way back in 2010. But the real success would be what happens to the reproductive status of the plant. When we look at the maturity linked traits, that is the single seed weight or the seed weight per spike, which is the major crop parameters, you will see the percentage reduction in the non-transgenics is 50% or so. But in the transgenics, it's about 20 to 25% or so. So we could cut the loss because of drought and salinity by 50% or so with these. But these transgenics, again, have we been able to give it to the farmers? No, because of a biotech policy issues. And today, we dream that we would be able to transfer them. That is my wishful dream. But we are utilizing this material for doing other pure science-based inquisitiveness as to what happens when you introduce in one gene. How does that gene interact in a different environment and so on. But that was the time when we shifted to another major problem of wheat, and that is of heat stress. Now, heat stress is one factor. With global warming everywhere, it seems that average temperatures are going to rise by 5 to 6 degrees in the coming decade or so. But in an immediate past, in the year 2006 or so, when we started this work, a year earlier to that, this region in India had higher temperatures as compared to the earlier season, and the temperatures were more than 5 degrees higher. Now, why this mere 5 degrees temperature increase is a matter of worry? It's a matter of worry because this increase in temperature occurred in the months of February to March, occurred very early. And it was not 5 degrees here, it was 7 degrees above normal. And the 7 degrees created a havoc because every degree which increases in temperature brings down your productivity by 4%. And for wheat, because it's a temperate crop, it is about 6%. So you can do your calculations. See, 7 degrees increase, 6% loss per degree, about nearly 50% of the crop yield was lost to heat stress, only because of heat stress. I'm not talking of other losses. And that is why, after a long, long time, in 2006, India had to import wheat. Fortunately, that situation has not arisen. We have not had to import wheat, but it was a good wake-up call for us to understand how to make heat tolerant. But before we could do that, we needed to understand what causes heat tolerance, what contributes to heat tolerance. There are no genes known for heat stress tolerance and otherwise. So we had to start from the ground stage. And when we look at the different growth stages of wheat, what we are talking of is Right now we're talking of the heading stage. Okay, February to March is critical because it is here that the spike emerges from the vegetative plant. Okay, and that is where you have about 40% loss in yield if heat stress occurs. But in the plains, heat stress, plains meaning in Madhya Pradesh, Maharashtra, even northern regions of Andhra and others, you will see that the heat stress is occurring when the plants are still in the vegetative stage of tillering, where the tillers are increasing. That means we realized that whatever would be the method of tolerance at the heading stage and the tillering stage, they will not be the same. So you need to address the two stages, two developmental stages very differently. Simultaneously, since fertility is also compromised, we wanted to investigate also what is the effect on the reproductive tissue. And in case of wheat, of course, the anthers and the carpels. So we took them also. Again, what the approach we took at that time was of the transcriptomics, and internationally also at that point of time, the viable project which was occurring was on understanding the expressed portion of the wheat genome, because wheat genome is huge and has lots of redundant DNA. So it was thought that the first two objectives would be on EST production of wheat. This is the international objectives I'm defining, okay? And you had to create cDNA libraries, do the screening, do the scoring, and then do the mapping, do the functional genomics also. Understand how the genome has evolved, how is it structurally capable. And then lots of bioinformatics also would have contributed to this information. So we went ahead, created the cDNA libraries after giving the heat stress, analyzed the transcriptome profile of the cell, and what do we see? Okay, again, I'm not giving you the experimental details, I'm just giving you the end product here. We saw that lots of transcription factors were getting upregulated after heat stress. There were lots of post-transcriptional control mechanisms in place. There were translational checkpoints. There were post-transcriptional control mechanisms in play. And there were many other things, etc. also, which are available. In fact, the whole set cycle, the cellular machinery gets reorganized to tolerate the heat stress here. And at every major step here at the molecular level, we needed to understand as to how heat is affecting these processes in different ways. So again, a very, very long decade or so effort. Lots of heat shock proteins have been characterized in detail in our lab, and especially the HSP26, the STI. We published this work in different journals. We have a couple of patents also. We worked on various transcription factors also, like prime being the master regulator, HSF-A2D, the zinc finger transcription factor, MADS-box, VZIP, many others also, and many intracellular signal transducers also, like the BRI, the B2, the vascular protein, the lipid transfer proteins, the STIs, some major osmolytes also, like the MEPS, myoinositol phosphate synthase, miscellaneous others which are associated with photosynthetic activity or with RNAi and epigenetic control mechanisms, and published extensively because of the detailed work we've done in characterizing each and every gene. But despite this extensive in-depth work, we asked the basic question, and we could not still, the world also still do not know what is the way how plants measure the temperature. What is the plant thermometer? We do not know. There are a couple of ideas about it, but none of them is universally accepted, and we still do not know how plants cope with temperature. Why? We have different things here. So what we realized at the end of the day was that besides answering some of the ways in which the genes were operating, we were opening up new questions to be addressed. And number one being, what is the plant sensor in the plants? And in the Indian context, when you go to the farmers in the field, they are not bothered about how many publications you have, what you have characterized the gene mechanisms in detail. They will ask you some very basic questions like, where the wheat, where the heat stress condition. And once the heat has come in the field, what can we do to negate the effect of this heat stress? Can we recover a plant? What should we do? So from their perspective also, we've tried to investigate how we can integrate our information, our knowledge, and see how the farmer can address these problems. So we took hold of all these 13, 16 wheat genotypes, which are popular Indian varieties, some tolerant, the others, and calculated their heat susceptibility index based on different parameters, based on the photosynthetic activity in the mature seed or the seedling stage or at the tillering stage. And we tried to see what are the membrane potentials, how the membrane stability changes, how is the chlorophyll changing, how is the thermal induction response changing, how is carbon being partitioned, how are the osmolytes, proline and others, and we calculated the heat susceptibility index. And we could measure this and we could say that this variety is more tolerant to heat as compared to this one. This is the most sensitive varieties which are tolerant, which are not tolerant to heat. And the variety which we were working upon, HD2329 and PPW343, which are the ruling varieties of wheat, are highly susceptible to heat stress compared to others like C306, which is relatively tolerant to heat stress. We also were looking at other genes, and while we were doing that, we came across one gene when we overexpressed it. If in a model plant that is like a Arabidopsis, you can see if the wild type cilic size is like this, when you overexpress it, the cilic size increases, the cilic length increases, the number of cilics per plant increases, and the seed weight increases. That means these are traits which contribute to increase in seed size and seed weight. And we are trying to incorporate this gene into wheat as well. We've done it in rice, but not in wheat. Then we also took hold of these two relatively heat stress tolerant varieties and two susceptible varieties. And this is how the grain looks at maturity. When it faces heat stress for about 2-3 days in the field, see the effect. This grain becomes like this. This grain becomes like this. This one, look at the sensitive, how it looks like. That is that they are most affected. So what do we do about it? So the BRI, which we had seen earlier, which was capable of increasing seed size and seed weight, that's a very expensive compound. But on a regulated way, when we spray it, we see that there is some degree of recovery. Now you compare this with this with this. See, it has nearly doubled in size, very closer to the control plants in the tolerant varieties. Little less in the susceptible ones, but still much better from here to here. There is about 30 to 40% recovery. We could use that. So we're looking at alternative methods, whether in the field also we could do something to rescue the plant's yield after heat stress. This is one such process. We are trying to study many other heat adaptive traits. Now, what are heat adaptive traits? Heat adaptive traits are ones which will lead to, say, photoprotection, like leaf morphology, wax, the pigments, the radiation use efficiency, how good light energy is being used, like carbon fixation, rubiscoactivity or rubiscoactivase, spike photosynthesis, because the spike also is green in color, it has chlorophyll, that can also contribute to the photosynthase. We are looking at membrane thermostability, we're looking at respiration, we're looking at how the carbohydrate is partitioned during and after heat stress, pollen sterility, meiosis, survival, grain filling enzymes, and how light is intercepted also, how efficiently water is used. To give you just a glimpse, I mentioned this RCA, rubisco activase. Okay, this is one enzyme which is very important. We have been able to locate an allele which is not sensitive to heat stress, and we have raised the transgenics in rice. Rice also you can see under the expression. But if you look at the photosynthetic efficiency of the rice plants, the light green is when the heat stress is given to the plant. Okay, it drops down, nearly 40% drop. But in the transgenics, it doesn't drop. That means this allele, which is tolerant to heat stress, when you overexpress that in rice, it can lead to heat stress tolerance in rice as well. So this work we are trying to translate into something which is more effective. Since rice is easier to transform, we are characterizing it in rice, but the ultimate goal is to do it in wheat as well. This is in case of the reproductive status also. You can see this is the seed set, which is about only 40% when the heat stress occurs in rice also. But in some of the transgenic lines, it is relatively better. It is about 60 to 70% or so. We still need to do further characterization, and this is how the inflorescences and the seed, the grains look like when the grains have seen heat stress or not seen heat stress also. We have tried to check for the fertility of the florets, seen how much sterility is there, what is the fertility percentage, and you can see the fertility percentage increasing from 40 to 20% also in some of the lines. And only the one need to be taken further. Similarly for grain weight also, how the grain weight is increasing or decreasing under heat stress also. Now, while we have been able to contribute quite a bit towards the heat stress, understanding the mechanism of the activation, the stabilization of the master regulators and the other transcription factors and other effectors, also the expression of different chaperones and enzymes, all these are associated with acquisition of thermotolerance. There is a whole different mechanism totally operative that is associated with heat stress memory. And this actually involves microRNAs, siRNAs, histone modifications, chromatin remodeling, lots of epigenetic changes and others, which we still have not been able to characterize completely. It takes time. We've done a little bit of work on epigenetics and the microRNAs also, but this heat stress memory is something which will give long-term adaptation to heat stress. So the mechanism of actions would be different for long-term and for acquisition, that is short-term heat thermotolerance as well. Of course, the major question remains is how this heat stress can be sensed, but we are still working towards this particular end. And hopefully, someday someone will be able to make sense out of the work we've been able to do. So this is where I would like to stop here, but I would definitely like to give you a message, both which I've learned from mulberry as well as wheat. From mulberry specifically, there was a time in Chinese history, if you see, silk was reserved only for the royalty. Common man could not wear silk. And how did we know about silk? It is your observation part. It is the Chinese princess who is credited with discovery of silk. How? She was sitting under a mulberry tree, it seems, having a cup of tea when a cocoon fell into her teacup. And when she took out that cocoon, she saw that a thread was emerging out of it. Okay. So the observation is when you put this silk cocoon in hot water, you can take out a thread. And the other significant observation is one cocoon has one silk thread. And that is, this two observations has what has given rise to the entire silk industry. Because the cocoon has one silk, you can get the thread out of it, you boil the cocoons, take the thread out of it, and only then you can reel the silk and make the silk fabric. So I believe it is your knowledge which is power in today's time. But your observation is the most key, and how you translate your observation into knowledge, how you do that is important. And today, since we live in a world of genomics, I think we need to have sufficient information how genes are working in a particular environment, how we have different genomic information, how we can exploit this information from one system to the other system and put it to good use for what we want to do. So I believe in the end, I would say observation is key and knowledge is what will give you the power to control what can be done and what cannot be done. So I would like to take your leave here. Thank you all for your attention. And I would love to clarify some queries if you have and if time permits. Thank you very much.
Thank you so much, ma'am, for this wonderful presentation. We are grateful for the time and effort you took to share your knowledge and ideas with us. That was really interesting. So now we will go ahead and take some time for a question answer session. Just a reminder, please be sure to type your questions in the question box in the control panel. Okay, so it looks like we have a question up.
Okay.
The first one, how do drought-resistant plants take up the nutrition?
Taking up the nutrition is not different. No different mechanism is involved. Only thing is lesser is taken up. Okay, because when water is less, it affects the transport processes, mechanisms of all other processes as well. So it's a water levels control how efficiently other things work, and that is what gets affected. And that is the whole purpose, to see that transport and cellular stability is maintained under drought conditions, under water stress conditions, because they are the ones which gets affected immediately, and that helps it. Otherwise, there is no purpose of doing this. I hope I've answered that question. Any other? That is the whole point. We want to make plants which can work even under limited conditions, water limiting conditions. Yeah. Any other question you have?
Okay. The next question here comes. Has the mechanism for water sensing identified in plants?
I would say yes, but it is not just one mechanism. There are different mechanisms present here. So different plants will respond differently, and different tissues will also respond differently. Primarily, it is the roots which will respond to water stress, and they have a different mechanism of operation depending upon which plant species it is. Then the leaves will respond differently, because it is there that evaporation occurs, and how water stress will influence it, and how evaporation is regulated under water stress conditions also. So these are specific terms here, and multiple mechanisms are in play, so that if one, and they work at different levels, I would say. They are folds of the same thing. So that is what it is, I would say. Yeah, any other question?
Yes, ma'am. So here goes the next one. Is there any climate resilience crop varieties that's developed till yet? And if yes, are they rich in nutrients?
When we say climate resilient, we mean which can withstand different conditions. Yes, I would say mulberries, transgenic mulberries, which we have developed, they will withstand drought, salinity, and we have tested heat and cold tolerance also. So they can grow ideally under normal conditions, but under stress conditions also they can grow. So they can grow under different climate conditions, and it is very important because mulberry is a plantation crop. So once you plant it, it should be capable of withstanding under different conditions. And we have tested for 4 major variables, abiotic stress. In fact, we have mulberries which are tolerant to biotic stressors also, against different pests and pathogens also. We have them with us, we have tested them also. So yes, we have plants which can withstand the climatic vagaries also.
Yes, ma'am. How much yield penalty due to drought in wheat?
What I showed you was about if it is normal, it could vary from anywhere from 30 to 40%. Depends. Okay, that is quite substantial only because of heat. We're not talking of infection and other things. So it is quite a bit easily 40% or so because of heat stress. And when you have the transgenics, there the yield penalty gets reduced. I would not say it goes off totally. So that 40% now reduces to 20% or so. At least 50% can definitely be recovered with the transgenics. So it's a relative tolerance, I would say, and relative gain of yield which is there.
Okay, here goes the next.
Yeah.
Since transgenics are not permitted to grow in the field at present, can we shift to gene editing to develop climate-proof crops?
We can always do that, but remember, even gene editing is type of a transgenic technology, isn't it? Because you're doing something, you're fiddling with the genome. So it will also have to go through the biotech safety protocols. It is not that you will be able to do it. Technologies are all there. What you should do is you should have more liberal guidelines where you can permit the use of these transgenics. I think that is what is more important.
What about the oil crops? Is there any transformance studies related to drought tolerance?
Oh yes, lots of them. Brassica is a major crop plant, which is oil producer. And there are lots of transgenics which have been created there for disease resistance, for abiotic stress tolerance as well. Yeah, it's there. You have the technology is available, you have the transgenics. Only thing what is lacking is the support to take the transgenics to the field, I would say. And sometimes it can be very depressing, you do all this effort and you cannot take it to the field. Imagine if the mulberry was allowed to be given to the farmers, because mulberry is not used for human consumption. It is the silkworm who eats the mulberry leaves. It's a vegetative, the plant in the vegetative stages can be cultivated because you constantly keep pruning it. So there won't be a problem, but it is not taken on a case-to-case basis. If BT cotton can be taken up, why not mulberry? But it is not there because there are no private players, no one is interested in the returns. But the public funded money, I think, should be with the public. If you allow it to grow it in wastelands where you cannot grow any other plant, suppose you're able to cultivate it, it's a beautiful source of fodder and feed also to many others. Okay? Yeah, any other?
Yes, ma'am.
Yeah.
So, moving from wheat to millets, is it sustainable? Grasses demand more water or less?
Grasses do not demand more water, I would say. In fact, less. And shifting to millets, I don't know what the person wants to ask. You cannot replace rice and wheat with millet. Millets can be added, can be substituted, because millets have a natural tendency, they are naturally tolerant to drought. Okay, so you can grow them in regions. See, the basic thing is agro management. Agri practices need to be managed. Where water is less, you can grow millets. Where water is more, grow rice. Where water is mediocre, with cold temperatures, grow wheat. That is what we used to be doing earlier. But now, with everyone wanting to do everything, that is where the challenge comes up. And you're not only ruining things, but you're ruining the groundwater also. You're using lots of fertilizer and pesticides, and you're contaminating the groundwater. That is the major cause of our. And most of these fertilizers are not biodegradable also. They will stay in the water system for pretty long time. That should be a major cause of concern. Yeah, other questions?
Yes, ma'am.
Yeah.
Is there any difference in the nutritional quality of the transgenic plants which are produced with respect to the human consumption?
Until and unless you have altered for the quality of protein or carbohydrate, I don't think there should be any. But again, it needs to be tested for each and every trait, I would say, each and every plant. I cannot give a generalized statement on that. Definitely not. But I do not anticipate that it would be different. Until unless I've targeted for, say, carbohydrate partitioning. It's only the process of partitioning which is being targeted, not the quality. There are certain reports where they've tried to manipulate the protein content also and increase the quality of what is there. Those cases are different, okay? And it is not necessarily to have the equivalent. Sometimes you can have more also. With transgenics, you can get a better quality as well. If your transport and other processes are high, then not only it is, one is quality of the protein and other is the quantity of the protein. Both things are important and you need to assess them also. Yeah.
Yes, ma'am. So we have the next one. Is there any alterations in quality parameter of crop by abiotic stress?
Yeah. Any other?
Yes, ma'am. Okay. Yeah. Is there any alterations in the quality parameter of crop by abiotic stress?
Usually, no. We don't suspect any, we don't anticipate any quality parameter to be altered under abiotic stresses. No. The quantity of the protein or whatever is a nutrient there, if it's a carbohydrate product or other things, that can be reduced, but not otherwise.
Yes. So far, how many transgenic plant varieties are waiting for a permission?
Huge, lots of them. And now people have stopped making transgenics because the regulatory processes do not allow it. We were stuck in between when we already had it and we could not take it further. But in India, no, it is not being allowed. Lots of them. You name a crop plant and we have transgenics waiting for, basically for crop plants, soil plants, vegetables, potatoes, not only for drought and abiotic stresses, but also for biotic stresses. You do have lots of them. I don't have the exact number with me, but easily I would say 40 plus would be the case in India. In India.
Are there any studies on associative microflora like rhizosphere or phytosphere in natural and transgenic plants?
Yes, there are work. There are lots of work which has been done on that. In fact, we have also tried to do that. I have not published that as yet, so I did not include that. We have also tried to see if, no, not on the transgenics. I've tried to see if microbes can help heat stress tolerance also. You know, there are lots of reports here. You need to check it, you need to validate it. But associated microflora in natural versus transgenics, yes, there have been work. I'm not able to recollect that as of it right now, but not our own work. But yes, there have been internationally, yes, people have taken care. In fact, when the biosafety regulations are laid down, this is one of the major parameters which they check for in the field as to how the microflora, if or not it is getting altered. Yes.
Does wheat and barley have hydroponic or aeroponic opportunity?
No, not at all. Why should you think of hydroponics? Means they're growing in water. Why you don't have water? Growing in hydroponics is possible only where water is abundant. You know, you're growing in pure water. We are talking when water is not there. That's a wrong notion altogether. Okay, and you cannot have wheat and rice growing in water because these are long-term crop plants. But these are not water plants. No. Certainly not for long terms. And you should not even think about it. I think it's a flawed question. You cannot have crop plants growing in as aeroponics or others. You can have other things here, maybe algae or no, that's not the right question.
Yes, ma'am. So, wheat or millets, which is better, taking into consideration health benefits?
Both, both. Have one exclusive of the. I would say take wheat, take millets also. Both of them. You know, in your balanced diet, the concept of your balanced diet comes into action. You should have a little bit of everything. That is what is the whole thing. I would never advise anyone, and no dietitian advisors skip this until unless you have a major intolerance to some major problem, like gluten. Only for those people, they recommend don't take wheat, take millets, but otherwise, a good combination of the two is always welcome. That is more effective, more nutritious.
Okay, so here comes the next. As he was discussing about the mulberry, there are even muga, tusser, and eri. Is there any tolerancy characters which are released?
See, the Muga, Tussar, Iri, they are very different organisms. The worm is different. The plant on which they feed is different. What I'm talking of is the mulberry. This is Mori culture. This is Bombyx mori. Okay, this is mulberry silk. In tussar, the the worm is different and it feeds on the tree species. Same is the case for tussar and eri. And you cannot cultivate muga, tussar, or eri. Whereas in the bombex, you can. It's a small scale industry. You know, in the sheds and so, the farmers will rear the silkworms. You can do it. So you cut the leaves and you feed it to the worms, and you can multiply the worms. But in the others, you cannot do that. Very limitedly only you can do that. Most of it is done in the natural conditions where it occurs on the tree. Okay, so the cultivation practice is different. The plants are different. The plants on which they feed also, the worms are different, and what they feed on is also very different. And it is very specific. In fact, that's a whole another new area of study, how different some plants are specifically eat only one type of plant and not others. Oh, I've raised quite a few questions. Good. But I'm glad that means someone has been following the lecture and trying to understand what I'm saying. Good. Yeah. Anything else?
Yes, ma'am. I think we have covered all our questions, and it comes to the end of our question answer session. Thank you, everyone, for putting up. So, ma'am, is there anything else you wanted to cover before I wrap up?
Me? Are you asking me?
Yeah. Like, how was your experience? Just a few words.
My experience working with the plants, I think, has been very challenging, has been very difficult, because we've not worked on only model organisms, we've worked on plants which are of importance to the Indian continent, and that is very important to take a problem head on, and we've tried to decipher each and everything one by one. So yes, that is a major challenge, but it is not an easy task at all. Good 30-40 years of work has gone in. When you present it in such a short time, every slide has the work of at least 5 years put in. So, but it is very satisfying at the end, I would say. I am very happy, very satisfied with what I've been able to accomplish, I would say. Both the plants are very tough. Tree species, people do not touch tree species. Wheat, people do not want to work on wheat because of their complex genome. They try to work on rice and other simpler plant species.
Thank
and
you
try
so
to
much,
constantly keep yourself updated with the technology, see what new technology is providing, and you need to be able to modify that to your own system. It's no small task. It takes a lot of effort. Some 10 years of work must have gone into developing the first wheat transgenics and so forth. It's not easy. It's a long drawn out game.
Yeah. Yeah, it's really challenging.
So that is why at the end of the day, it can be very challenging. It is very disappointing also when at the end of the day, when you have something ready to be given to the field and your policies do not allow it to.
Yeah.
So it's that kind of a situation. So imagine all your antibodies, all your vaccines, everything is transgenic, chimeric, just man-made. You allow that, isn't it?
Yes.
So one needs to be a little open-minded about it, And even all your haploids, not haploids, all your hybrids, they're also man-created, no? So you allow that. Their whole genomes are mixing, and you're not allowing one gene to be introduced. So it is ignorance which is leading to all this opposition, I would say. And that is that's a wire mechanism. When you say gene editing se kaam ho jayega, no. Gene editing is also you're tinkering with the DNA. You don't know what all effect it would have. It will also have to undergo the same protocols, same biosafety measures. So there is no short-term mechanism which will give you thing. You have to just manage your policies in such a way to do it or not to do it. Otherwise, the government should stop giving funding for this kind of work if they don't want to take it to the ultimate, to the field. I would say that, when it is government funded, then the product should be given to the field. And who are our policymakers who don't understand what you're doing? See, that's the pity of it. The one who doesn't know, he is taking decisions on the subject which he has absolutely no inkling about. Anyway, it can enter into a debate. I don't want to rake up that right now. But I feel I'm very passionate about my work. I feel bad that we have products lined up with us and we are not able to give it to the release.
That was really nice of you, ma'am. Thank you for sharing your experience and for your wonderful contribution.
Thank you, sir.
So now, to conclude, I welcome Serene Sarah ma'am to express a vote of thanks.
Yes. Good afternoon, all. Today we have listened to yet another fruitful session on climate Smart Agriculture in the Era of Plant Genomics by Dr. Paramjit Khurana, University of Delhi. She gave an elaborate, detailed perspective about the plant genomics and climate smart agriculture methods, the changing phase of agriculture world over, and the efficient utilization of land, water, bioresources. So, and the management of the ecosystem for a sustainable livelihood. And also, she explained clearly about the era of genomics and the genomics, that is what is happened and what can happen, what is going to happen, everything she uh, explained clearly to the participants. And also she discussed about the research activities like sequencing of rice, wheat, tomato, mulberry genome, mapping, etc. And I think the participants have benefited much. You have clearly explained in this vast subject, and then in this 1 hour time, 1.5 hour time, you have given a vast idea about, you have shared the valuable knowledge. As you rightly said, knowledge is power and observations will count. That is a key point. So I think ma'am has clearly conveyed all the ideas regarding the plant genomics and its aspects. And thank you, ma'am, for sharing your valuable experience, valuable activities, your collaborations, etc. And also, I wish you good luck for all your new endeavors. Thank you so much, ma'am. And I place my extreme gratitude to IFSA, Indian Women Scientists Association, for organizing this program in association with BRNS, Government of India. Without their help, this program must not have been happened or materialized. As Rita ma'am rightly said, she also told that let's work together for a better tomorrow. So we all will work together for a better tomorrow. In fact, we have been constantly associated with IFSA, and that centenary webinar series, one of our centenary webinar series was also sponsored by IFSA, and looking forward to have more association with IFSA in our future activities also. And my thanks goes to, thank you Rita ma'am so much, and thank you all the IFSA members also, Surekha ma'am and all. Thank you so much. And with our my thanks goes to my principal, Dr. Tara K. Simon, and manager in their absence for their support and encouragement for all the academic activities. And also, I would thank my mentor, and my extreme gratitude goes to my mentor and our adjunct faculty, Dr. Susan Epen. She is the guiding light in our department. She always gives us the best. As I always say, she gives us the best by giving the best resource person. And in this popular ongoing popular lecture series, she provided us the best. And thank you, Susan ma'am, on the behalf of the department and on the behalf of the college. Thank you, ma'am. And I thank Shyam sir, our head of the department, for his constant support and guidance extended in all our academic activities and also in the successful conduct of this webinar. And I also thank my students, my faculty members, and my students who gave this technical support, especially Fatima, Ashwin, Inosh, etc. And this, without their technical support, this program would not have been a great success. And I, last but not the least, I thank my participants around the globe. I saw international participants also. So I thank all of them for spending their valuable time in this during this program throughout the program. And last, I'm telling you that we are also going to conduct a webinar on the trends in modern biological science, from developmental biology to metabolic engineering, that is sponsored by National Academy of Sciences Kerala chapter, and it is scheduled to conduct on 18 September 2021, and hope to see you all during this next webinar also. That is at 10:30 same time. So see you all during the next webinar also. And I see the Dr. Edathil Vijayan, Dr. KV Peter, etc. also in this webinar. Thank you all the participants, and thank you sirs for being presented here. By then, stay safe, stay healthy. Thank you all. Thank you very much. Thank you, ma'am.
Thank you so much, Paramjit, for this lecture and the connectivity
you, sir.
and all the thought processes which crossed the mind as you spoke.
Thank you so much.
Thank you, Paramjit.
Thank you, Susan.
Bye.
It's a pleasure to have you.
Thank you, man. Thank you so much.
Thank you for the excellent presentation. Very, very nice. And thank you.
Mr. Paramjit Andapan.
Yes, yes.
Okay, two Paramjits are there.
Yeah.
Okay, it was an excellent lecture.
Very, very good. Very good. Very good.
I think Sunita Mahajan also was there. Sureka could not attend. She had some other.
Yes, yes, yes.
എന്താ എന്നാ